Modern biology textbooks categorize umami as the fifth basic taste, but this classification rests on a widespread psychophysical fallacy. For 3 millennia, taste classification was strictly subjective. A basic taste had to be perceptually independent, meaning it could not be replicated by mixing other tastes, and it occupied a distinct sensory space in the mind. This classic paradigm reflects an evolutionary survival mechanism. The immediate identification of sweet energy or bitter toxins serves as a rapid oral detection system, allowing for a split-second decision to swallow or expectorate. This model shifted in 2002 when advances in molecular biology isolated the specific receptors responsible for detecting umami. Armed with anatomical proof, the scientific community hastily added it to the list of basic tastes. This creates a critical academic tension. Ligand binding at the molecular level is not a biological guarantee of a distinct, perceptually independent flavor profile. When subjected to formal psychophysical criteria, umami fails the fundamental requirement of perceptual independence. To identify exactly where the current model breaks, we must subject umami to the 7 strict criteria used to define a basic taste. A receptor-ligand complex is biologically significant, but it is psychophysically insufficient to define a basic taste. The evidence requires a complete taxonomic overhaul. This decision tree outlines the 7 criteria for basic taste classification, beginning with an evolutionary advantage and a distinct class of effective stimuli. Umami clears the stimuli requirement through the existence of free L-glutamate, found naturally in tomatoes, seaweeds, and aged cheeses. While pure L-glutamate generates a weak response, it becomes robust when combined with ribonucleotides like IMP and GMP, the synergy responsible for the flavor profile of cured meats. The next requirements concern anatomy, the presence of specific transduction mechanisms, and neurotransmission pathways. Umami possesses these specific receptors, primarily the T1R1/T1R3 heterodimer and several MGLUR variants. Its electrical signals are successfully carried to the brain via the chorda tympani and glossopharyngeal cranial nerves. However, testing human perception is difficult because isolated glutamic acid is actually sour. Eliciting a perceived umami response requires the acid to be bound to a cation. Clinical testing utilizes monosodium glutamate, or MSG, to provide this necessary cation. While umami passes every anatomical hurdle, this chemical reliance on sodium causes a critical failure during human perception testing. This brings us to the critical node of perceptual independence, the requirement that a taste cannot be confused with any other basic quality. To test this, researchers measure hypotasters, individuals with a measurable reduction in their ability to recognize specific stimuli. Clinical testing data shows that approximately 27% of the population are classified as umami hypotasters. These subjects may reach a detection threshold, sensing that a substance is present, without reaching a recognition threshold where they can identify what it is. This chart shows the overlapping recognition thresholds of hypotasters. At a 29 millimolar concentration, these subjects cannot reliably discriminate between MSG and simple table salt. For over a quarter of the population, the L-glutamate is effectively invisible. They are exclusively perceiving the sodium cation. Substituting sodium with potassium, or MPG, fails to solve the problem, as potassium introduces confounding salty, bitter, and metallic sensory noise. This is a reproducible limitation inherent to the molecular makeup of umami stimuli. Sweet, sour, salty, and bitter exhibit no comparable level of population-wide sensory confusion at these concentrations. Because 27% of humans cannot isolate umami from salt, it fails the perceptual independence benchmark. Umami also fails through a second cross-modal overlap with sweet tastes. This factor model compares the molecular architecture of the 2 receptors. The umami receptor is a heterodimer pairing T1R1 and T1R3. The sweet receptor relies on that exact same T1R3 subunit, paired instead with a secondary protein. This physical overlap correlates with behavioral data. Individuals with poor umami discrimination frequently show lowered sweet taste sensitivity. This line graph shows dietary intervention results. Prolonged consumption of MST causes a simultaneous parallel decline in the intensity perception of both umami and sweet stimuli. This suggests that umami peptides interact with sweet receptive domains, preventing clean perceptual isolation in the brain. These molecular and behavioral overlaps strip umami of the distinct boundaries required to function as an immediate pre-ingestive survival signal. If umami lacks the perceptual clarity to dictate rapid decisions in the mouth, we must identify its actual evolutionary function. L-glutamate detection begins with saliva release, but its primary action occurs further down the alimentary canal. The T1R1 and T1R3 receptors are not just in the mouth, they're expressed extensively throughout the lining of the human gastrointestinal tract. Within the gut, the detection of L-glutamate acts as an endocrinological trigger. This diagram tracks the release of cholecystokinin, or CCK. Binding L-glutamate in the gut initiates a cascade that releases this hormone to regulate protein digestion and gastric motility. This release directly influences behavioral outcomes. Meals preloaded with MSG and IMP significantly increase post-meal satiety. This satiety effect occurs completely independent of the actual protein or carbohydrate content of the food. Umami functions as an anticipatory signal, using flavor detection to prime the digestive system for the breakdown of complex nutrients. Its utility operates on a delayed gut-brain axis feedback loop, fundamentally separating its role from the immediate oral alerts of the classic basic tastes. Based on its lack of perceptual independence, umami definitively fails the diagnostic test for a classic basic taste. Continually adding newly discovered receptor ligands, like kokumi, fat, or calcium, to the basic list ignores physiological reality. This matrix illustrates a proposed solution, splitting the taxonomy to establish a distinct subclass called alimentary tastes. Basic tastes are defined as strict pre-ingestive alert systems for rapid oral decision-making. Alimentary tastes are post-ingestive metabolic primers, detecting nutrients to coordinate digestion, absorption, and satiety. This classification system resolves the receptor fallacy, acknowledging molecular discoveries without forcing them into an outdated framework. Umami serves as the case study for this shift, moving from an anomalous basic taste to the prototypical alimentary taste. Reclassifying umami corrects a widespread misconception and provides a physiologically accurate model for the future